so people can refer to it. You're asking for what? Human tissue?
Yeah, I see that you did something that says human tissue, Ethan, and then you have it like mapped to colors.
Oh, yeah, I made that as you're looking in whatever
Color scheme.
the color thing is. I'll share my screen. I have that open. Right here. I'm on strain right now. But right here. I did this only because I needed tissue colors for my plot. That's the only reason I did it. And
Yeah.
you'll notice like down here they're kind of ambiguous. Like I made the brain just all shades of green.
Hmm.
So yeah, are you doing something else?
So the idea is, let's see what Yang said exactly. So we have like a list of like all the cell types in like with and they're like respect. Sorry, it's all the GSMs and their cell types listed with it.
Yeah. I'm confused because I try to give Yang this, and this is something I did a while ago. It's this awk script because this annotates all of the branch IDs. But I guess are you guys doing like tissue instead of branch ID?
Um, yeah, basically that, but for tissue. Yeah.
Okay.
So yeah, I was gonna go and annotate the data.
Okay.
At least that's what I interpreted what I was doing as. I mean, but yeah.
Okay. So what did he put you in charge of the colors?
Um, I don't know. I
Okay.
was just gonna find a way to like match it with the cell types given.
Oh, that's easy. All you have to do is you just use you literally use a single function. Shoot, give me a second. It's right here. It's read Excel, and then you do this dollar. Sorry, I'll put it in the Slack. And this creates the mapping that you're probably wondering about.
And that's in units. Just the read Excel, or is that it looks like
That's an
a
R.
yeah. Okay, that's what I thought.
And all it does is it creates a mapping between like the hex and the tissue. So for instance, here, it's going to look at the tissue column, and then it's going to map to the hex. And then you can use it in a function like this. So if I look at where tissue color is used, like for my usage, I'm using a function that creates a summarized experiment. And a summarized experiment, you can kind of think of it like an Excel document where it has the data and then it has like row data and then column data. So for instance, in the spreadsheet right here, the data is actually these 1s, 0s, and 2s. The column data is what's in the columns here above each of these colored rows. And then the row data is what's on the left-hand side with the tissue description, TBK, and sample. And then that function maps that color to this tissue column here. So you never really, like, you don't, like, it's made that way so that you don't have to put the hex in the sample sheet because you can always do the mapping. But then
Yeah, I see.
you need harmonized, um, you need these to be harmonized across cell types, across each sample. And if Yang's giving that to you, I would look off of like what I did in awk. Basically, here's like how I kept track of it here. I would define the branch IDs on the left, and then I'd iteratively go through and look for which keywords fit in that category. So B cell, this dot means any character, so like a space, and then the question mark means it's optional. So it could be B cell, B dash cell, B dot cell. $$ cell. And then you can use awk to do matching. And you're saying if the line contains glia, mark it as glia. That's
Yeah,
what.
I was gonna do something along those lines, but in obviously in R.
Yeah, it
But
makes
I it'd probably be easier in just with using awk now that at least now that I know awk better.
it's easier in awk because for the reason that it's easier to move files around, because otherwise you're gonna have to like load an RDS file in, uh,
Yeah,
process it, and then export it.
yeah, that could be a pain. It is a pain
I know.
from my experience working in R
To
with
stay
that.
out of R as long as possible. But now I'm back in R doing analysis. Cool.
I like R. It's
Yeah.
it's it's like familiar, you know.
Yeah.
That's
So
why I like it.
yeah, you can do it however which way you want. Um, so did he tell you to just do the tissue stuff?
Looks like it.
Okay.
Excuse
So
me.
then the tissues that we defined, I don't know if you want to take, I don't know if you like I don't know if you want to reference the branch IDs, but maybe the tissues you should go after first would be those that are in the color document. Um, Not under mine, under this one, because these are like actual tissues. These are kind of like tissues and cell types.
Hmm.
But he included some blood. So yeah, just start with these 25 or whatever, 28.
Okay.
Cool.
I think he wants me to add like colors depending on like new encountered cell types.
Yeah, so then just do your best to make it like, okay, so let's say you come across some new tissue or some new, like that's what I did. I had monocytes, neutrophils, granulocytes I added here, and I just made it a similar color. I made it this reddish pink color, and then I also had to add fallopian tubes and ovary cells, so I just added a blue that went with testes. And then yeah, I mean, Wanding's probably going to go back and change these colors a little bit, but the idea is to make the colors kind of make the difference in color like akin to the difference in cell type. So if cells are close together, you want the colors to be close together.
And is this just for graphic purposes? Like
Yeah, it is. It's
graphic
literally
have,
it's literally for like this legend right here on the right hand side.
so it's just all consistent, I guess.
Yeah. So in each
That's
paper
fine.
and each figure we write for each paper and figure we create, it'll be the same.
That's good.
And then the same thing can be said about strain right here.
Hmm.
It's just a different tab in here. Cool. Yeah. Yeah. Good luck.
Yeah, I'll see what I could do. I've been busy because
I got
obviously
you.
you know this final weeks in Drexel are
Yeah, I
nuts.
know how it goes.
So, oh
Cool.
yeah, thanks.
You got it.
Good luck with your finals
Thank you.
and exams. I understand.
Yeah, it's like 3 more weeks, right? So
I don't even
Yeah,
know.
we're finishing up week 8, and I think week 6 to 10 are the worst.
Midterms are week 2 to week 10.
The entire term is midterms,
Yep.
basically.
How
So
it goes.
honestly, yeah, like usually until week 5, I don't feel any stress. But then like once it hits week 5, and then after, it's just like a roller coaster downhill.
yeah, I mean, I feel like disconnected from school, so I feel like I'm stressed, but not at the same time. I don't
Yeah,
know.
I
It's
can
okay.
relate.
It's definitely more stressful in person from
Yeah.
my past experience.
But
Being
that's
remote is kind of relieving in a way.
very true.
Huh, funny. Cool. All right. Uh, you have any more questions, Mike?
No, I guess I'll just like refer to Yang. Anything
Okay.
else? I got a good idea though of what
Okay.
is
Cool. And
expected.
then the document I showed you in awk is on um, on the lab journal.
Lab journal.
Yeah.
Okay, what's it called?
Oh, what is it actually called?
Yeah, because you probably have a lot.
Yeah. One sec.
2021.
Sample sheet underscore branch ID underscore curation. I can copy and paste it into.
Huh, can't find it. You said it's under lab journal and under your name, I guess.
Yeah, under 2021.
Under 2021.
Under reconstruction. It's like halfway down.
Oh, under
Oh, actually, no,
reconstruction.
it's not.
Oh,
Yeah,
okay. All right. And what was it again? Branch ID
branch
creation?
ID curation.
Awesome. All right. Ooh, look at that. If statement.
Yeah,
That's so exciting. All
all
right.
cool.
Thanks.
Yep. All right, Elaine.
Hi. I'll turn my camera on. Hi.
Hello.
Um, so, okay, so I have this ginormous spreadsheet and I'm trying, oh, bye, Michael. Um, I have this spreadsheet and I had trouble just putting it into normal Excel. So I'm on Google Sheets right now, but I know I shouldn't.
You want to share your screen?
Sure. Okay. So here's my enormous spreadsheet. And so I sorted it based on here from A to Z.
Okay.
So there's a bunch of cell lines with just numbers, but then like obviously when you scroll down more, there's all the other ones and like for instance, like
Is
I
this
code.
okay? Is this? Can you go to the left?
Yes.
What does he want you to do with this?
He told me to organize the spreadsheet, find like the characterized cell lines, and then implement the geo data, the VAF to the geo data.
Okay.
I think
So do you want to do this in a little faster way? Is is this file on HPC?
probably.
Okay, then let's go find it. So can you connect to HPC?
Okay. Should I? Okay. Should I do
I
this?
will show you how to. I will show you how to do all of this in code.
Oh, nice.
I
Should I
know.
connect to this?
I can't see your. I can't see what you're pointing at, but I would assume. I
Oh,
would assume. I would assume.
I mean the whole. There's a lot of stuff on my desktop right now, so just ignore that. That's what I was talking about, the VPN.
Yeah. Okay. Um, so it's not on. Are you gonna? You're gonna freeze a little bit. Welcome back.
Okay. Um, so this is very weird. Okay, so I just connected to the VPN. Did you say it's not an HPC?
But I'll put it there.
Hmm, okay.
Okay. Can you open Linux and log on to HPC?
Didn't we write this somewhere? Haha, found
in your
it.
notes. I can save it as a command because I do SSHZ for Zhou.
Okay. Okay. Hello. Here. Do I just do that?
Yes.
What is my password for my laptop? Oh.
Okay. So I want you to. Okay. So I want you to. I'm just going to show you the command I'm running to move it over to HPC. So are you on Slack? Yeah. Okay. So in the random channel, this is what I'm using. It's called SCP, and it's copying it from the Dropbox, and it's going to move it over to sample sheet. It won't work for you because you don't have Dropbox. It might work for you, but it's with my IP address and my username.
Okay.
But now, if you go to the sample sheets folder, do ls. Let's see if it's there. Let's see if you have sample sheets are sent. Do not. Delete desktop documents, downloads, music, pictures, public templates, videos. Delete the ones in like dark blue.
How do I delete them?
It's rm dash rf and what you want to delete. Space between r and dash rf. Dash rf or command line arguments, command line arguments followed by the dash command.
Did I still do it wrong?
Oh yeah, sorry. It's rm space dash no space in between r and f. The dash indicates command line arguments, and then space, and then desktop, and then case
Yes.
sensitive.
Am I supposed to?
I don't know why I did that. Yes, you've removed the desktop. Now you have to do it again for other ones.
Is that for me? My typing might be very loud.
It's fine.
In public too.
Yeah, that's the worst part.
So did it work? Let's see. Wow.
On.
All gone. Do I take any others out?
Yeah, templates and videos. You just don't need them. I just wanted you to kill time. So
Okay.
okay, so we're gonna create a symlink that points to the lab sheets, which is where I copied the file to. So what you're gonna do is you're gonna go on Slack and you're gonna go on the random channel, and what ln-s does is it creates a symlink. So you're gonna copy that, and that's gonna basically point sample sheets to lab sample sheets in the Zhou _ lab folder. Do that. Okay, and now you do ls. And now we have sample sheets. So you can cd, you can change directory to sample sheet. Do
To
like
sample
S,
sheets?
do S and then tab. Does that actually not work? Okay, then type out sample sheets. I don't know. Okay. And then you're gonna have to go to 2021. So CD to 2021. Okay. Yes. Okay, and then do LS. Yeah, so here we are. These are all of the sample sheets. And you can see that yours is in there somewhere. You'll probably see your name. Yeah, Elaine, that one. So what we're gonna do is we're gonna use a command called xl2tsv.r. And the way you use that is you're going to first off, I want to, can you do which and then type that command space and then which space and then the command. It's XL to TSV because it's an Excel file to a TSV. No Excel file. Okay, so we have to add that in. So give me a second. Okay, can you do, you'll learn it easier. Can you do this? I sent it in Slack. So we're opening up something called the bash RC folder, and that should have information about okay. So the way you kind of, okay, so let's see. You can change your command, you click I to insert. So it changes the mode to insert, right? And then what you can do is you can add this line that I sent to you at the bottom of the file.
Where do I add it?
The bottom of the file. Oh, click escape. Okay, you could do that too. Click escape and then click lowercase o. Okay, and then hit paste. Then do control V or whatever. Okay, there you go. And now you can click escape. And to save the file, you do colon WQ for save and quit. So colon. You have to do a colon. You're not doing a colon?
I don't know.
Okay. Colon? It's colon. That.
I was putting it in, I swear.
WQ. And now you saved. And then now to reload this, what you do is you use source.bashrc. Oh, you have colors. This is so cool.
Fun.
I know. So now if you do which, I think that'll work. Yeah, okay, so what that thing just did is it loaded a file, which is kind of like a bash RC. So a bash RC is a file that's loaded every time you log in. So then these are like your options. These are like what your profile looks like. And Wanding has particular options laid out in a bash RC that should be shared by all the members. So what it does is it adds coloring like this. It adds something. It adds some something in his lab bin, and in this lab bin is this right here, and this is what we're going to use. So the way you use it is you use the command, and then the argument is a TSV is a Excel file, and then you can pipe that to less, and then you can view the contents. So you want to give it a try?
Okay.
Okay, so you do excel to tsv.r, that's the command. I don't know why your the tab completion doesn't work.
I feel like it usually does.
Okay, and then space, you need an option, and that option is going to be the name of the file. So do 2021. So it's going to be the file in that Google spreadsheets that's right behind you. or okay, you can look up too. So it should be
Right
wherever
there.
you see a lane, right
Right
there.
there.
Do you see that right there?
Yes.
Okay, and then pipe that to less.
Just put enough.
You have to use the pipe. If you didn't use the pipe, then Excel to
Touching?
TSV would read that. Yeah, Excel to TSV would have read that as another command line argument. Yes, but piping it moves the output of the command to the input of less. Yes, so do that. And what should happen is you should see skipping, just wait. Okay, and this is the magic. You can see it now on Linux. Okay, and you can use the arrows to move left and right. Yeah.
Where's cell line?
So you see cell line right here?
Mm-hmm.
Okay. So we need to figure out what column number that is. Right? So, oh, okay. So you have, do we want to count or do we want to do something programmatically so I can teach you something? Oh, it's
One,
right there. Okay, you want to count?
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 15.
15. Okay. So let's play around with this a little bit. Let's
Okay.
figure out, it's going to be annoying if we have to keep reloading it. So what we're going to do is we're going to just make a temp. Let's just do this.
Yeah, I
Basically,
was about to ask, like, how do I get here?
sorry.
Like, how do I get here usually, just if I want to work on this again?
Well, look where you are. You're in the sample sheets 221 folder, and that's where you are.
Okay.
Yeah. Um, let's actually make this a little more friendly. Do we have anything open? What are we gonna do? We're gonna save it to the lab journal. You're
Amazing.
working on something. 2021. That was explicit sniffs, and now what we're gonna do is we're gonna do 2021 05 20. And what's the name? What was the name of this? Okay. So what are we doing here? We are, let's do EF for Elaine Ficara. And then what are we doing here? We're doing cell line. Um, actually, don't ask me why, but I want to do geo cell line dot sh because it's going to be a bash script. Okay. So now what we can say is we can say, okay, where are we? We can say, okay, we can do echo pwd, and you get here by doing, oh, not by doing that. I know you're tired. You get it by doing that, and then what we can say is we can do this command, and that command is going to, right, it's going to convert it into a TSV file. Sorry. There you go. It's going to convert it into a TSV file, and you can either pipe that to less like this, or you can direct the output to a file. So what we're going to say is we're going to say, okay, we just want to turn this into a TSV file. So that's what we're going to do. So we don't have to we don't have to run this over and over again. We can just play with the TSV file. Right? I can't hear you. You are off. Somebody turned her off. Turn her off.
I was trying to say I need my cursor to unmute myself.
Oh, that's hilarious. Just keep yourself unmuted.
Sorry, I muted myself because my laptop's overheating.
It's fine. Okay.
But I had a question. Oh, what exactly is a TSV file?
Tab separated value. Have you heard of a
Tab,
CSV?
a CSV, yeah.
It's a comma separated value file. It just means that the values are separated by commas.
So that's what that means. Okay,
And a
cool.
TSV means that you're separated by tabs. You can have a PSV, which is something I made up, which is pipe separated value.
You made TSV up?
I made PSV up in a workshop I did a while ago. You could do QSV for quotation mark separated value. It's just an acronym. Right?
Okay.
So now we can view this file, right? And this is the file. Did you figure out what line? Figure out what column cell line is in?
Wasn't it 15?
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay, it is 15.
I can count.
So what is wanting want you to do now that we have this data, right? This is our data. What is wanting want you to do? He wants you to figure out the
From my understanding, there's a lot of cell lines that don't have info, and the whole geodata needed to be like sorted or organized, and then just like we want to look at the characterized cell lines.
so the ones that are most popular.
Yeah, and implement VAF on that.
Okay. So let me just be a Snoopy Snoop. Oh, genotype information. So did you go on encode?
I went on Encode, but I've never been on Encode before, so all I did was search the cell line and see what came up. And like some of them, like the ones that were not like popular, commonly used, had like no data, while the ones that are more commonly used had a lot of results. But I mean, I don't know what I was like supposed to look for.
Oh, let's figure this out then. Obviously, it's like, ooh, encode. Oh, you clicked that one.
I did. I used
Well,
it.
I'm following the breadcrumbs.
I've been here.
You've been here.
hmm
So how would you figure out which one has genode information?
Well, I went to data type. Can I use it? I went to go to data type, arrow down, and I looked at data set. I don't know if that was like where I was supposed to go, but I was like, oh, maybe that means like they might have some information about like the data itself. I don't know.
Um, I wouldn't know how you'd quantify that.
I mean, he wasn't sure about using encode. It was an idea of his.
So why do you need genomic information? Why do you need genotype information? What does that mean?
Isn't it to proof of concept? Like, um, I thought like we need. Okay, so the goal of this project is to ensure that participants have genomic privacy,
Hmm.
and so I think he was saying how they don't want to be identified by like their age, sex, status of disease, etc. And so I guess like also maybe like cell line identity, like I'm not, I don't know. Well, I guess maybe something I need to make sure I know is how to implement VAF on that. Like I guess that's the whole
Yeah.
right.
So what I was gonna do, and what I thought that he meant, is to look at the cell lines to which we have the most data, and I
Exactly.
was gonna do
Go to Gila. There's a bunch. There's a Gila of five.
okay. But what you did is you sorted. What did you do? You sorted the Google Sheets file
A to
the
Z.
by the cell line.
I
So
sell
you can
line.
do that in here by doing sort F15 and then typing this to less,
Mm,
and then I'll do the same thing.
amazing.
It gave me an error. Am I imagining things?
Maybe.
No, F is ignore case. Numeric sort. Just why am I forgetting how to sort things? Give me a second. Oh, that's bad. Oh, somebody just got. Sorry. Okay. Yeah. It's a 15. Okay, and now it's sorted by cell line. So if you go over here to 15, it's hard to see now, isn't it? An easy way to just isolate the 15th column is to do that. Just cut 15th column out. So
Oh.
right now, see, we have a bunch of NAs,
Yeah.
and obviously we don't want those.
Hmm.
So what you can do is you can. Do you understand what I did here?
Yeah. You just isolated the 15th column, which was cell
Yeah.
lines. Only look at that.
So what you can do is you can grab all of those. Uh, you can grab all of those columns, grab all the rows that aren't NA. That's something that you might want to do, right? That don't have that in your that don't have that in the 15th column. So you can do that using grep, and grep has this character called dash v, which means invert. So if I do grep dash v na, it'll return to me all of those that don't contain NA, but I only want to apply that to the um, a single column.
Hmm.
So I have to figure out how to do that. Um, let's what I do. Here we go. Someone has the same question. Saying use awk, which is what I was going to do.
Yeah.
This is awk as well. So, all right, grab. What we're going to do is awk instead, and we're going to say if 15 doesn't equal
Mm.
15 doesn't equal NA, and we're going to print the whole row. Right?
Okay.
Does that make sense?
Yeah.
Okay. And what we can do, we print it here, and we can type this to sort dash F15. and then cut different columns.
Sheesh. Okay.
Got invalids in there because sort takes basically the column parameter is different and okay,
Why are all the NAs there?
because I said if it doesn't equal NA, but the problem is that it doesn't equal NA. It equals NA in quotes. Okay, so to make it easier, what I'm gonna do is I'm gonna use this tilde, which means regular expression, and then I'm gonna say like a regular expression is kind of like a substring. So what I did is I'm saying if in line 15 there isn't NA, print a line. If sorry, if in column 15 there isn't NA, print a line. Then instead you get this.
Okay, that's better.
And then what you want to do is you want to do unique dash C, and these will basically give you the count of all of them. Okay. And what you can do is you can sort by the first column. Okay, you do dash K to indicate column number one, first column on the left, and then N means numeric sort. And you do this, and let's do a reverse so that you get the highest number. Like that. So this is basically the data. This is what the data looks like in respect to cell lines.
Okay, so this is a much more digestible version of the data. Can you explain the sort K1 and R again?
Yeah, yeah. So so these dashes. Remember when you did this?
Yeah.
Basically, you have the command. So Linux always expects the first thing you type to be a command. So arm is a command, and then spaces afterward indicate command line arguments. Okay, and then typically when you want to specify a type of command line argument, you use a dash, and then the name of the command line argument. So R for RM dash R, the dash R in RM means recursive, so it'll delete subdirectories. And then F means that you want to delete the file. So you can either use them together or separated like this, but people often just do it like this because it's less space. So in sort, you could split it up like this, and then we do the same thing. Dash K means you're trying to sort by a column, and then what follows K is the column one. Dash N means you want to sort by a number instead of lexicographically. When you sort lexicographically, that means that you're sorting by an alphabet, right? So what would happen is that um, 10 would come before 2 because 1 comes before 2. Sorry, 10 would come before. So, okay, I can just show this. That'd be easier to show you. You see this?
Mm-hmm.
See how 86 comes before 76? Actually, wait a minute. It should happen like that. Hmm. I guess you don't need the N then. I guess it automatically does it. Anyway, the N means if you do sort dash dash help, it says the N means numeric sort, compare according to string numerical value. So you sorting according to numbers instead of strings. And then R means reverse, so instead of doing ascending, it does descending. So now you understand that these are the top however many cell lines in the data.
Oh, that makes sense. Huh.
Yeah. So, okay. So another question you might have is, okay, so how do you apply the VAF onto the data, right?
Exactly.
How would you do that? So let's just dig into it.
Okay.
Dig into it. So where did Wanting say the data is located? In here.
Yeah.
So this is the directory that we want to look in. So let's get out of here. Let's just do say understand the data by number of
That's not how you spell
I'm using for my course. I've always been bad at spelling. It's been actually very bad for a long period of time.
it.
So now we want to do is perform
I can tell.
perform VAF on one singular cell line sample. Let's just do one sample. Do that. Okay.
Okay.
So let's look at the first sample in the spreadsheet. So you do that. What we're going to do is we're going to go here. All right. First sample, that's HM450. Actually, wait, that's not a good way to do it. We have to do something else. We want to ask, okay, which, if the second row is HM450, then the entire row. Simply one, then the entire row. Because if we had a bunch of if we had a bunch of HM27s, we wouldn't be able to see, we wouldn't know how far down HM450 is, and now we won't have epic in either. And right, we're accessing data only from HM450. So let's take this first one right here.
Sorry, I realized my light was off this whole time.
And then what we want to do is we want to say, so those are IDATs, aren't they?
Yes.
Hmm. Ooh, that's real good. I just want to see what the files look like. Because I imagine these come in pairs, right? So when you run this function, you should expect to, you have to pass in a SID set. What you're going to have to run is something like this. Or rather, what you're going to run is the read IDAT pair right here. That generates a SIG set. You should expect to use this. You should clean this up eventually because these arrows right here are from merges using GitHub. So you can clean those up later. You see like head right here? They're from merging with GitHub. So yeah. So now we're gonna say in R, you're gonna run this to get a big set, and then going to effectively run this function. So let's say E S N M S that E Smith and I Smith. Okay. And you're gonna send it to Sigma. Like this. That make sense? Okay.
Okay.
You have a question? I mean, I'm not done yet, but do you understand what I'm doing? I'm using this function read IDAP pair. Well, we're not in R, so I can't. You won't actually feel the errors I'm making. You have to trust me. We do that,
Yes.
and then
Okay.
this, you have to actually send it a. Uh, you have to actually send it the. You could give it a. What read IDAP pair gets is not a directory, a file directory. We have to get that, and that's why I'm trying to see what kind of data is in here because I actually don't know what the data looks like. I
Mm-hmm.
actually want to see like what the from that. So
No.
I just want to look at it. Does that make sense what I'm doing though?
Yes. Yes.
I don't know how to like this directory is so large. I don't know how to look at the files in it.
Mm,
Oh,
okay. I'm applying this to IDATs, so why did I make the cell lines? I'm still confused on that.
I think he wants you to apply it on cell lines first. Let's try to do something. Let's try to do something easier. I don't know if this is gonna work at all. Find. Okay, so find is another. Oh, that's bad. Find is another command that is helpful.
Does
And
sorry, does caret C like stop something from?
yeah, it stops it.
Okay, that's good to know.
Yeah, if anything's going out of line, just use Command C. Okay. So find is a command that allows you to kind of look in a directory and see if you match like file names or like find files that satisfy a certain condition. That's what find does. So you can do something like find, and then I want to say that contain this. Ooh, actually, I don't know if it'll work. Does it work? I don't know. Find file name. That okay. Linux find file name that contains. Find max depth name. There it is. So we have to do it like this. Putting quotes. I'm hoping that the IDAPs are identified by the GSM. That is what I'm hoping. Otherwise, I'm not sure. Ooh, I'm not sure what we're gonna do. Okay. because I don't know how we can view the files in this directory because it's probably so large. I actually might do some hunting on my own to figure this out because it's ridiculously large. LS. Oh, actually, yeah, I doubt it'll find anything, but the idea is it's going to give you a directory. It's going to give you, it should give you two files that contain this GSM. One file is going to be an IDAT from the green channel, and then the other file is going to be an IDAT from the red channel. And then you give that to the read IDOT pair, and then that's what you send in to get implicit and get explicit stuff. That should not be the same function. Right. Also, something to note is that if you were to call this function without doing anything before it, you get like a function does not exist. What you have to do is you have to take this right here, and I would just put it like right here.
I learned from experience. I need to do that. Yeah.
Yeah, you need to define it.
I need to define it. I've
Yeah.
learned.
Now that it's defined, you can use it. But do you understand what I'm doing right here with the find? I'm trying to figure out what file name is identified by this GSM.
No, actually, I don't know what the GSM is.
The GSM, so GEO, you know GEO is right.
Yeah.
The GSM is a GEO accession. It uniquely identifies either a data set or I don't know, a data set or a sample. So this GSM uniquely identifies this. What is it? Is it a sample? Yeah, it's a sample. And this one so happens to be, you know, so we use GSMs because they're unique, we use them to identify the samples. So that's why you see them in sample sheet because they can be directly linked to GEO. So, right, in the Excel spreadsheet, which we turned into a TSV file, on the left-hand side, it had the GEO accessions. I picked the first one, and I'm trying to figure out which file, which IDAT in this directory contains this GSM. So then um, you know what the files are. But I have a second thought. Source name, platform. No, it doesn't have it in there, does it? Okay. Yeah. So I doubt it'll load, but I just want this. Oh, man, I'm also running something on my end because then what I did is I'm just trying to list all of the files in that directory because I'm just generally curious how they're formatted because I have no idea what they look like. But yeah, cool. Okay.
Well, I
Yeah,
learned something for sure, but I have
okay,
some questions I'll make sure to ask Wanding tomorrow.
sure. Do you have any questions for me?
Um, mm, no, I think you answered all of them.
Yeah. Eventually you're going to put some path in here. Path to GSM air or something like that.
Okay.
And so like if you find out that like in that directory it's green and then red, all you'd pass in to this is the path but with the GSM.
Okay.
You wouldn't include the green or red because it knows to add that on because this path indicates that there is a red and green channel there.
Okay. That makes sense. And whenever you do print, um, $0,
But that
it's
means the entire line.
and then and what does the Excel to TSV do again?
This converts an Excel file. So an Excel file is a binary file. It converts it to a text file that's separated by tabs. It's the TSV.
Okay. 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 Yeah, I'm good.
Yeah, I'll let you know if my thing finishes because then I'll have a better idea of what the IDATs look like what the file names look like in that folder. But I'm almost positive they're by the GSM. Look at all this information. Look at all this. Look at all these steps.
Learned a lot.
They're
Okay.
not. Don't think of them as steps. Think of them as small problems. You just solve one and you go to the next one.
Okay. I I love how the line 8, if you just do so much in one line.
Yeah, it's because you just have to manipulate data, and this is all just. This isn't even to do anything. It's just to look at the data.
Yeah. So the awk is letting you just isolate the column 15 and only read out the ones that are without NA.
Yes.
And sort by column 15, only show column 15. What was unique? C?
Unique. So what unique does is it removes duplicates when you give it an ordered list. So if I were to give it without the -c flag, it would just return all the unique. It would only return each cell line once. When I give it the -c flag, if it returns it returns the counts of the unique classes. So that's how I can see the number. The number. That's a good question. I didn't explain that.
You can see the most
Yeah,
used cell lines in the data. Okay, that makes sense.
and then I sort it to see them in descending order. Yeah.
What do I need to figure out again if the
We need
fine
to figure out. So what we're doing is we're picking a GSM here, like we're just picking the sample GSM, and we need to figure out how that GSM maps to an IDAT file stored in this directory. And we don't know that because we can't view the directory because there's so many files in that directory.
GSM is a what does it stand for again?
GEO I don't know. It's a unique identifier. It's like an accession number. It's like in code has accession numbers. This is like an accession number for a sample.
Or something. Okay.
It could mean GEO series matrix or something. I don't know.
Okay.
Cool. All right.
And I would use that instead of an IDAT.
No. So this GSM maps to this row, and this data corresponds to this GSM, which is from GEO. this GSM also has respective IDAT files. So the idea is that this kind of acts as a key that maps the IDAT files to this row of data.
Oh, okay.
Yeah,
That makes sense.
yeah,
Okay, thank you.
yeah. And then what's going to be cool, and which is a step I wanted to do, is when you can use the data in here, like you do this filtering, and then you use the first row. So you use the first column of GSMs, and then you view, and then you use that, and you send that data to the read IDAT pair because you know how it maps to the IDATs in this directory.
Okay.
That'd be the next. That'd be the next step. But right now we have to figure out how to do it for one, a simple case, one. Cool. All right. Well, that's that.
Thank you.
You're welcome. I will see you later.
Okay, I'll see you. Bye.
Bye bye.